Welcome to Virology Conversations, where we break down complex topics into digestible discussions. We take carefully curated scientific sources on a virology topic and generate AI podcast-style conversations. Think of this as your companion for better understanding the most complex topics. In this conversation, we're exploring how viruses outsmart one of our most powerful defenses, the interferon system, and how these battles determine which species a virus can infect.
Welcome to the deep dive. Today we're tackling something really fundamental to how new diseases emerge: cross-species viral transmission. When we hear about outbreaks, maybe SARS-CoV-2 or Nipah, the focus is often on how the virus gets into the cell, finding the right receptor. But that, as our sources this week make clear, that's really just the first step, isn't it?
It really is. Getting in, that's the initial challenge.
Yeah.
But the real make or break moment, the thing that determines if it's just a dead end for the virus or, a major outbreak, that often comes down to whether the virus can handle the host's immediate superpotent defense system. We're talking about the interferon system, or IFN.
Right, the IFN system. It's like the cell's panic button designed to shut everything down almost instantly when a virus shows up. So our mission in this deep dive is to unpack this amazing molecular arms race. And we're going to focus on something quite startling: the idea that the barrier stopping a virus from jumping between species, say from a bat to a human, can sometimes boil down to just one or two tiny changes, like one or two amino acids in a single host protein.
Exactly. It's incredible sensitivity. We've looked at a couple of really comprehensive reviews here, detailing how this IFN system gets triggered, how the signals work, and crucially, how viruses fight back. It's this constant evolutionary battle fought at the molecular level that dictates which species of virus can actually infect.
Okay, Love IFN, phase one, the host side of the story. Setting up this antiviral alarm. What is this IFN system, fundamentally?
Well, think of it as the body's first responder against viruses.
Right.
Extremely powerful. There are different types, but the main ones we focus on for direct antiviral action are type I and type 3 IFNs.
Type I, that's alpha and beta interferon.
Correct. Type IFNs are sort of the broad spectrum alarm. They don't just trigger antiviral defenses in the infected cell and its neighbors. They also ramp up the wider immune system, activating killer cells and so on. Type 3, or lambda interferon, is also antiviral, very much so, but its effects are more localized.
More targeted.
Yeah, its receptors are mostly found on epithelial surfaces. Think the lining of your gut, your lungs, places where viruses often first try to get a foothold.
So the response is tailored. But how does any cell know it's time to sound the alarm in the first place? How does it spot the difference between its own stuff and a virus?
It looks for things that scream foreign invader. These are called pathogen-associated molecular patterns, or PAMPs. And the cell has detectors, pattern recognition receptors, PRRs, specifically designed to find these PAMPs.
And for viruses, especially RNA viruses, a key PMP is double-stranded RNA.
That's a big one, yes. Historically, dsRNA was seen as the primary trigger. Healthy human cells don't typically have long stretches of dsRNA floating around. But RNA viruses, whether they're negative strand or positive strand, inevitably generate dsRNA during their replication or transcription cycles. It's a byproduct they can't easily avoid.
Okay, so the cell sees dsRNA and knows something's wrong. Where are these detectors, these PRRs, located?
They're positioned strategically. You've got sensors inside internal compartments called endosomes. These are the toll-like receptors, or TLRs. TLR3, for instance, sits there waiting to detect dsRNA that might have been brought into the cell. TLR7 looks for single-stranded RNA, and it's especially abundant in certain immune cells.
And then you have sensors right out in the main workspace of the cell, the cytoplasm.
Exactly, because that's where many viruses replicate their RNA, so you need detectors there too. These are mainly RNA helicases called RIG-I and MDA5.
RIG-I is fascinating, isn't it? The way it recognizes that specific chemical tag.
It really is. RIG-I specifically latches onto RNA molecules that have a 5' triphosphate end. See, our own cellular RNA gets that triphosphate cap modified very quickly after it's made. So if RIG-I sees an RNA with that raw 5' triphosphate, it's almost certainly looking at viral RNA, a very clever distinction.
That is clever. Okay, so a sensor, maybe RIG-I or a TLR, detects the viral PAMP. What happens then? How does that signal actually turn on the interferon gene?
It triggers a signaling cascade. Think of it like a chain reaction inside the cell, ultimately leading to the activation of specific proteins called transcription factors. These transcription factors then need to assemble on the promoter region of the IFN beta gene. Researchers call this assembly the enhansome.
They enhance them like a molecular construction crew.
Sort of, yeah. You need several key workers to come together, IRF3 and F-kappa B, and another complex called C-gene and TF2. They all have to bind near the gene, often working together.
And is one of them the absolute essential one?
IRF3, or its close relative IRF7, is indispensable. Without IRF3 or IRF7 binding, you don't get IFN production.
How do those factors get activated? I know NF-kappa B is normally held inactive, right?
That's right. NF-kappa B is usually kept tied up in the cytoplasm by an inhibitor protein called I-kappa B. The viral detection signals lead to the destruction of I-kappa. Once it's free, NF-kappa B can move into the nucleus. At the same time, IRF3 gets activated through phosphorylation. Chemical tags get added to it. This causes IRF3 molecules to pair up, forming a dimer, which then also travels to the nucleus. When they all meet up at the IFN gene, boom, the alarm is sounded loud and clear.
Okay, phase 2 then. The alarm is going off. The host cell is mobilizing. But viruses aren't passive victims here. They fight back. How do they sabotage this alarm system?
Oh, they have a whole arsenal of tricks. A primary strategy is to cut the lines of communication right at the start. Target the adapter proteins that relay the signal from the sensors.
Like cutting the phone lines.
Exactly. Take hepatitis C virus, HCV. There's a protein called NS34A, which acts like molecular scissors. It specifically cuts TRIAF, which is the adapter for the TLR3 sensor. And crucially, it also cuts a protein called MAVS.
MAVS is the adapter for the cytoplasmic sensors, RIG and MA5.
Yes. So HCV, with one protein, effectively disables signaling from both the endosomal and the cytoplasmic RNA sensors. It's a very efficient way to shut down the initial alarm.
That's surgically precise. What about other strategies, like hiding the evidence instead of cutting the wires?
That's another common tactic, sequestration. If the sensors can't find the PAMPs, they can't signal. So viruses like reovirus or influenza A, with its famous NS1 protein, produce proteins that literally bind to and hide the dsRNA produced during replication.
So the dsRNA is still there, but it's masked.
Right. It's coded by the viral protein, so RegI and NF5 can't detect it. This also has the side benefit of preventing dsRNA from activating other antiviral proteins downstream, like PKR and OAS.
We also see viruses going after the transcription factors themselves, sometimes in a very species-specific way.
Absolutely. That porcine delta coronavirus, PDHV, is a great example. Its N protein is specifically adapted to target the pig version of IRF7. It marks the pig IRF7 protein for destruction by the cell's own garbage disposal system, the proteasome. This shows how tuned these viral countermeasures can become to a specific host.
Okay, let's say the virus fails to stop the initial alarm. Interferon gets produced, signals to neighboring cells, and triggers the expression of hundreds of antiviral genes, the ISGs. These ISGs are the real executioners, setting up the antiviral state. How do viruses deal with them?
They have specific countermeasures for the major ISGs too. Let's hit the big three. First, PKR.
PKR, the protein kinase R, it senses dsRNA too, right?
Yes, and when activated, PKR phosphorylates a crucial factor for making proteins called EIF2 alpha. Phosphorylated EIF2 alpha shuts down almost all protein synthesis in the cell, host and viral.
A drastic measure. How do viruses stop PKR?
Different ways. Some, like pox viruses with their K3 protein, make a pseudosubstrate that basically tricks PKR. Others, like influenza's NS1 again, can directly inhibit PKR.
And herpes simplex virus, HSV1, has that really clever workaround with ICP34.5.
Ah, yes, that's quite elegant. Instead of blocking PKR itself, HSV1's ICP34.5 protein hijacks a cellular enzyme, a phosphatase. It redirects this phosphatase to specifically remove the phosphate group that PKR added to EIF2 alpha. So it reverses the shutdown, turning protein synthesis back on, conveniently allowing the virus to make its own proteins.
Sneaky. Okay, executioner number 2. OAS.
OAS stands for 2'5-oligodinylate synthetase. Like PKR, it's activated by dsRNA. Once active, OAS produces these small molecules called 2'5A. These 2'5A molecules then activate another enzyme, RNase L.
And RNase L is the shredder.
It's the shredder. RNase L chops up RNA indiscriminately. Viral RNA, host RNA, everything. Another drastic scorched earth tactic by the host. So viruses, again, can use proteins like NS1 to hide the dsRNA and prevent OAS activation in the first place. Or some viruses like HIV-1 or EMCV can actually trigger the host cell to produce its own inhibitor of RNase L, effectively disarming it.
Wow, okay, and the third major player, MX proteins.
MX proteins are different. They're large GT paces, kind of like molecular clamps or cages. They don't rely on dsRNA sensing in the same way. Instead, they often recognize specific viral structures, like the nucleocaps of the protein shell around the viral genome. Human MXA protein, for example, can recognize the nucleocapsid of viruses like nogotovirus and physically block it from getting into the nucleus, which stops its replication cycle cold.
So direct physical interference. And beyond these intracellular battles, viruses also have ways to evade the broader immune system surveillance that IFN normally ramps up.
Definitely. They need to hide from immune cells like T cells. HIV's UP protein is a classic example. It targets an ISG called BST2, or tetherin. Tetherin normally acts like a rope, literally tethering newly formed virus particles to the cell's surface, preventing their release. VQ disables tetherin, allowing HIV to escape efficiently.
And they mess with antigen presentation too.
Yes. Since interferons boost the display of viral fragments on the cell surface via MHC class, i.e. molecules flagging the cell for destruction, many viruses have evolved ways to pull those MHC molecules back inside or block their assembly. Anything to avoid being spotted by killer T cells.
This brings us to phase 3, and perhaps the most mind-bending part. How tiny differences, literally single amino acid changes, create these species barriers. We've seen viruses evolve antagonists highly specific to their host's proteins. What happens when they jump species?
That specificity becomes their Achilles heel. The viral protein might not recognize or bind the new host's version of the target protein effectively because the sequence is slightly different, and sometimes very slightly.
Let's revisit that HSV1 example with RIG-I.
Right. HSV1 has that UL37 protein, or the amidase. It targets human RIG-I, specifically hydrolyzing an asparagine amino acid at position 495 and 495. This modification inactivates human RIG-I.
But it doesn't work in mice.
It doesn't. Because mouse RIG-I at that exact same position doesn't have an asparagine. It has a lysine, K495. UL37 can't modify the lysine. And researchers prove this. They changed just that one amino acid in human RIG-I, swapping N495 for a lysine. Suddenly, human RIG-I became resistant to the HSV1 protein.
One amino acid. That defines a huge part of the species barrier for that virus.
It's a major factor, yes. A single-point mutation difference between humans and mice determines susceptibility to this specific viral evasion tactic.
And we see something similar with the DNA sensor CGAS and the same HSV1 protein.
We do. UL37 also targets human CGAS, deamidating it at residue N210, shutting it down. Now, interestingly, gorillas and mice also have N210, so they might be susceptible to this specific trick. But some other non-human primates have different amino acids there, making their CGAS less vulnerable to HSV1's UL37. Again, subtle variations matter.
Let's talk STAT2. That's a key signaling molecule downstream of the interferon receptor, and it seems critical for flaviviruses like dengue, Zika, yellow fever.
Absolutely critical. The NS5 protein from these flaviviruses is incredibly good at binding to human STAT2, HSTAT2, and targeting it for degradation. This completely shuts down the IFN signaling pathway in human cells.
But not in mouse cells.
Not in mouse cells. MouseStat2, MSTAT2, is resistant to being degraded by the flavirus NS5. And that difference is a major reason why these viruses don't typically cause severe disease in regular lab mice. You need special mice, ones that are immunocompromised or, more tellingly, mice engineered to have the human STAT2 gene humanized STAT2 mice to actually model the disease properly. It highlights STAT2 as a key host range determinant.
Even within really closely related species, like different types of hamsters, these tiny differences can pop up.
They can. Look at pox viruses and their PKR inhibitors, E3 and K3. These inhibitors are notoriously species-specific. Vaccinia virus K3, for example, works well against PKR from mice and horses, but it fails against PKR from humans, pigs, or Armenian hamsters. But then, comparing Armenian hamsters to Syrian hamsters, two very similar species researchers found that the ability of the virus to inhibit PKR effectively hinged on differences at just two amino acid positions in the hamster PKR protein, residues 463 and 464.
It's like this constant rapid-fire molecular arms race. A tiny change on one side forces a tiny change on the other. We see that with HIV and ApoBac3 too, right?
That's the absolute classic example of this co-evolutionary battle. ApoBac3G is a host protein that tries to hypermutate the HIV genome to death. HIV-1 evolved its VIF protein specifically to destroy human ApoBac3G, HA3G. But many monkeys have ApoBac3G versions that HIV-1 VIF can't degrade. And researchers found that sometimes the difference comes down to a single amino acid. If you change just one residue in human A3G position 128 from aspartic acid to lysine, D128K, it suddenly becomes resistant to HIV-1 VIF, mimicking the resistance seen naturally in African green monkeys.
One mutation flips the switch. It's incredible.
It shows how rapidly both host and virus can adapt. A single mutation in the host can provide resistance. A single mutation in the virus can overcome it.
Okay, phase 4. Given how fragile these barriers can be, resting on just a few molecules, what does this mean for us? For predicting the next pandemic, for developing treatments?
Well, first, it underscores how quickly viruses, particularly RNA viruses with their high mutation rates, can adapt. We saw this happen in real time with SARS-CoV-2. The alpha variant, for instance, became more transmissible, partly because it simply got better at suppressing the IFN response. It didn't necessarily evolve a brand new weapon, it just produced more of its existing IFN antagonists, like ORF9b and ORF6, compared to the original Wuhan strain. A quantitative change leading to a qualitative difference in outcome.
And this isn't new historically. Influenza has done this.
Oh, yes. Analysis shows that influenza A viruses independently evolved ways to overcome the restriction imposed by human MXA protein during both the 1918 pandemic and the 2009 H1N1 pandemic. The specific mutations were in the virus's nucleoprotein, the protein MXA targets, different mutations in different pandemics, but the same functional result. Better evasion of a key human defense, likely contributing to their pandemic success.
So if these barriers are so specific and adaptable, relying only on standard human cell lines in the lab might not give us the full picture for predicting risk, right? We need better models.
That's a huge point. We desperately need models that reflect the natural host reservoir. If a virus originates in bats, studying it only in human or mouse cells might miss crucial aspects of its interaction with the bat IFN system. This is why developing things like bat organoids, mini-organs grown in the lab, is so important. It lets us ask, why doesn't this virus cause disease in the bat?
How does knowing all this molecular detail actually help us fight viruses?
It informs strategy on multiple fronts. Take vaccines. If we know which viral proteins are essential for blocking the IFN response, we can specifically delete or mutate those genes to create live attenuated vaccines. The virus is still alive, so it provokes a strong immune response, but it can't effectively shut down the host defenses, so it remains weakened and safe. Many modern vaccine strategies use this principle.
Makes sense. And for antiviral drugs, same logic. Instead of just randomly screening compounds, we can rationally design drugs or peptides to specifically block the function of these viral IFN antagonists. If you can find a conserved region in, say, the paramyxovirus V proteins that's crucial for blocking M5 signaling, maybe you can develop one drug that hits that target across multiple dangerous paramyxoviruses, like Nipah and Hendra.
And then there's the really cool application, using viruses against cancer, oncolytic viruses.
Right. This directly exploits the IFN system. Many cancer cells, as part of becoming cancerous, develop defects in their own IFN signaling pathways. They can't sound the alarm properly. So you can engineer viruses, take VSV or HSV, for example, make them extra sensitive to interferon. In healthy cells with a working IFN system, the virus gets shut down immediately. But in the cancer cells with the broken IFN system, the virus replicates like crazy and kills the tumor cells. It's a beautiful therapeutic concept based entirely on this host-virus interaction.
So wrapping up, this deep dive really highlights the IFN system as this crucial double-edged sword, absolutely essential for surviving viral infections. But its predictable nature, the specific pathways and proteins involved, gives viruses a clear roadmap for evolving countermeasures.
And these countermeasures are often incredibly specific, finely tuned, sometimes down to that single amino acid interaction.
Exactly. The virus has to walk a tightrope, suppress IFN enough to replicate, but maybe not so much that it kills the host too quickly or triggers overwhelming inflammation. Sometimes viruses even seem to use aspects of the IFN response to establish long-term latent infections.
Which leads to a really provocative final thought. You mentioned chronic infections. If some viruses, like certain paramyxoviruses implicated in long-term illnesses, manage to persist by constantly dampening the IFN system, what are the hidden long-term consequences of that subtle, decades-long immune suppression or dysregulation?
That's a critical question. Could the persistent blocking of IFN signaling by a latent virus contribute to chronic diseases that we currently don't think of as infectious? There's speculation, for example, about links between persistent paramyxovirus infection, which we know are masters of IFN evasion, and chronic bone disorders like Paget's disease. It suggests that the ripple effects of disrupting this fundamental antiviral system might extend far beyond the initial infection, potentially manifesting years later as conditions that look entirely non-viral.
A fascinating and slightly unsettling thought about the long shadow cast by this invisible molecular warfare, something for all of us to think about. Thank you for joining us on the Deep Dive.